Excellent, and thank you everybody for the opportunity and I guess the energy at the end of the day to listen to this talk. So it's really a pleasure to be here and we're excited to be a part of this. Well, this is going to be about the spatial resolved transcriptomics for geomics spatially, digital spatial profiler. And Fadi Isi at the beginning of the day gave some really great information. And I'm going to kind of curtail on top of that and explain a few other things that I think could add on top of it. And then show some really interesting examples of great publications that have been done using the technology. I like to start with the fact that we started with a technology called nCounter, and it's a bulk gene expression platform. It became really popular because of its ability with FFPE tissue and difficult matrices like blood, and its ability to get data easily. And I think geomics is living up to that as well. The Geomics Digital Spatial Profiler was launched about a year and a half, almost 2 years ago. And it's now a tissue-based platform that is actually upstream from the nCounter. It can also, that if it has a particular probe for the nCounter, or it can then now have something for NGS platforms, depends on the level of plexing that you want to do. I think I'd like to at least just briefly say what's special about it at a really high level. This is now a spatial platform that is going to give you FFPE and fresh frozen. We can do this now. Everything here is doable now. We do RNA and protein. We have tunable ROI selection. And I've got quite a few slides that explain that because there's really big flexibility in being able to pick your ROIs and defining that early. We can do very high plex, 18,000 gene whole transcriptome for human, 22,000 gene whole transcriptome for mouse, and then 100+ plex protein if you want to validate that or you have translational aspirations. And then also throughput. If you've got a project that has a lot of slides, we can do a lot of slides in a day. And then the quality of the publications. We do really pride ourselves in people getting actual data and publishing these in really high impact journals. In just a year and a half, there's been 45 peer-reviewed publications to date. And I'll be going over some of those as well later. And of course, nature methods, we're all excited about that. And I think it's one of the reasons we're here. It's very cutting edge technology. We are now we are in this spatial space. And we launched the geomics, like I mentioned, a year and a half ago. And in 2022, we're launching the spatial molecular imager. We've got some data on it now. Don't want to go into too much detail, but it is a really interesting platform where there's very easy sample prep. It can do up to a couple hundred in protein now, and we're building that out, and then 1,000+ plex in RNA. It's going to be fully automated. It, of course, will come with the different analysis software packages that we will bring to the market. And this is just a visualization of what it can do. So imagine this is just visualizing 5 out of the 1,000. And FFPE, of course, we can do that, fresh frozen, just visualizing the locations of the different genes being expressed. And then we're going to be able to do it at high resolution, 2D and 3D as well. Like I mentioned, 1,000 plex, very high sensitivity, comparable to single molecule fish we feel as well, with really low copy number sensitivities. And then the FFPE, fresh frozen, and organoids. We've got some really interesting posters out now. We had a really good launch of this at, I think it was AGBT earlier this year. And a couple of those are out of the Fred Hutch and some really interesting CRISPR work being done at the Dana-Farber Cancer Institute, and a couple that we did showing the 1,000 plexing capabilities. So how does the DSP work? I'm not going to go into too much detail, but it's important to reiterate because Fadi went into this a lot earlier, but just in case, we have a photocleavable linker. So if you wanted to do protein work, this is attached to an antibody. Or if you wanted to do mRNA quantification, it's attached to a complementary sequence. When UV light hits the indexing oligo sequence gets released. And then this is what, in essence, then gets read either by the nCounter or an NGS system downstream. In this mix, we also will add fluorescently labeled antibodies or an RNA-scope-ish probe, frequently these antibodies though. And if you see a fluorescent signal, we're not using it for quantitation, we're just using it to help define where we're going to draw an ROI. And then you can see we're going to hit these photocleavable linkers with UV light, release those probes into solution, sip it up, read it downstream, and then the data comes back. And you have a lot of different visualization techniques. And of course, everything is brought back to your ROI. And the flexibility in the ROI is really key. So as far as how the technology works, this is it. We have a micro mirror array with 1,000 mirrors that are about 1 micron square in size. So the smallest size we can illuminate with UV light is 1 micron. And all of this is how do we shine UV light with this micromirror? And the next couple slides are nothing more than us releasing probes from exactly the part of the tissue you want it to come from. Example on the right here is a tumor example, pancytokeratin, labeled in yellow, and then the software sees a mask over pancytokeratin. These micromirrors trace UV light and only release probes in pancytokeratin. And you're enriching for that population of cells. And then we invert that mask and look at the stroma. What the software is actually doing is really is this, and I think this really drives it home. You have a beautiful crisp image with those fluorescently labeled antibodies. This one's antibodies, the morphology markers. And then you define a region of interest. In this example, they do a circle. And then the software with very simple image analysis. That's really also one of the beauties is the image analysis is, this is a microscope. It's actually a highly multiplex gene expression technology hidden in the microscope. So the image analysis is marginal. Very easy here where you're picking tumor versus non-tumor areas and illuminating that masked area against each other. We're illuminating the tumor area, getting those probes versus the non-tumor area. And then we're doing it all over the tissue. And then if you wanted to do it for a protein panel, you can do that for protein in tumor versus non-tumor. Or of course, we now have whole transcriptome capabilities. And if you wanted to illuminate everything in that ROI, you can, and you can get whole transcriptome for mixed tumor and immune cells, or for that type of segmentation. You can just do the tumor cells and look at their transcriptome, or you can just do the immune cells and just look at their transcriptome and compare these against each other. And then also very exciting is the ability to illuminate one cell type. So we have single cell population capabilities. Our micromirror can illuminate down to 1 micron. So we can very easily illuminate a single cell. On the left is 3 fluorescently labeled antibodies to show us where to illuminate our UV light. And then on the right, we are illuminating the tryptase mast cell markers. And we do like to get a pool. So we can get 1 cell population with about 10 to 20 cells for protein, and then mimicking what Fadi says, about 60 cells for RNA. That being said, we've certainly seen groups go down to 5 cells and still be able to detect 4,000 to 5,000 genes in just 5 cells. So there's definitely, you can push this envelope, and we give you the flexibility to be able to do that. We can do fresh frozen FFPE. Another nice addition is that we also allow for tissue microarrays. I frequently work with groups that are doing 1 to 50 sections of tissue on 1 slide, and you very much have the ability to do all that ROI selection on all of these tissues to really increase your biological replicates. And then of course we have software, and the software is easy to use. It allows you to go down to your gene, visualizes everything with heat maps and volcano plots and a lot of nice, easy to use features. And then with regards to the publications, I really want to kind of go into the science. And I want to go into the science for a few reasons. 1, it's the most important part. But 2, we're really proud of the publications that have come out. Groups are able to publish with our technology for a couple reasons. At the end of the day, our technology is an IHC or an ISH, and then we're releasing 18,000 genes or that 100-plex for the IHC. So we work with a lot of different tissue types and are really robust in postmortem tissue and FFPE. So I think that's one of the reasons people are able to publish so quickly. But then also, its data analysis is not trivial. And when you are able to annotate at the beginning of your process with an experimental question, then when you have your cell populations of tumor versus stroma and then 18,000 genes to compare against, the gene expression information is more approachable. Or one cell population, let's say there's a lot of brain information going around today, a population of just astrocytes around an Alzheimer's plaque, and you want to look at the whole transcriptome gene expression of that astrocyte population in a particular region of the brain. That's simplified by getting to that in the beginning. And one of the examples I think is really interesting was a group where they came to our Technology Access Program, which is our TAP program, to early access. And then within about 12 months, they got some really interesting data that they ran another quick experiment with us, and first got the Nature Medicine paper, and then 9 months later got a Nature publication. And that was a group out of MD Anderson. It is Dr. Jen Wargo's group, and the first experiment in the TAP was this was all with protein. The first experiment with the TAP, from what I've heard, they really were mostly interested in T cells, and that maybe B cells weren't as important, but our panel had it included. And then they certainly found some immune markers associated with response to immune checkpoint blockade, and then noticed that some of them were B cells. This was the Nature Medicine publication. 9 months later, they got the Nature publication. And here they decided to look at inside and outside of tertiary lymphoid structures. And they found that B cells organize into TLSs and melanoma and are a biomarker for response to immunotherapy. And they worked with Lund University. They were co-authors on each other's paper, and Joran Jonsson also found that B cells and TLSs synergize with killer T cells to target tumor cells. One thing that isn't as apparent in this particular publication, and it was mentioned at the beginning, the interest that we're moving into this cross-omics, that omics isn't just protein, isn't just RNA, isn't just DNA. We're now moving also, this group here overlaid an H&E image. And that is so at its core, they overlaid an image from another imaging platform to then drive ROI selection on our technology. And we are now working with Fluidigm groups with their Hyperion system to use a really highly multiplex 50 or 30, 40, 50 plex antibody panel to help drive ROI selections on our platform. If you're doing metabolomics, work with a mass spec imager, or lipidomics, work with a mass spec imager. We're working with different groups to have these imaging mass specs drive ROI selection on our platform. And I think that's really some exciting cross-omics and really pushing the envelope. Also, I wanted to bring out just some quick examples of, I think, difficult tissue types. So this is a group out of the University of Colorado, Phil Owen's group, which was looking at prostate cancer and had lytic and blastic bone metastasis. And you can see, they used our nCounter panel, or they used our nCounter bulk gene expression system. They used some of our protein panels as well, but our technology is robust enough to work with decalcification, and drew their ROIs on this really difficult sample matrices. So I'm just trying to drive home examples of different sample types that we can work with. And then I really like this example, another tissue type that they worked with. It was with small cell lung cancer. This was a 44-plex protein. And it was with a group out of Yale, David Rin's group. What I thought was really interesting here that I wanted to highlight, one, it's TMA, so they did many, many cores on one slide to increase their biological replicates. But then also on the left here, you see the fluorescently labeled antibodies to help drive ROI selection. And they, on the right, put a mask over the fluorescence, and you can see that they compartmentalized the tumor area, the CD45 positive area to CD68 positive areas, illuminated those separately, and then those separate illuminations could then be compared against each other in this sample, and then the reproducibility between slide to slide and the cores on the same slide. And then of course, they found, not of course, but what they ended up finding is that CD45 compartments, when in CD45 compartments, upregulation of CD4 and CD56 improved progression-free survival in these patients. But really, it's the TMA cores, biological replicates, the capabilities with that ROI selection. And then also, I thought it might be interesting to mention this colonic biopsy example, where this was the X-linked severe combined immunodeficiency, so XSCID, FFPE samples as well. But once again, for the sake of time, I'm not going to dive too much into it, but I really want to show the flexibility. So this was, I showed at the beginning of how we are able to illuminate UV light to draw these ROIs and get out the genes from or proteins from exactly where you want. In this example, they illuminated the mast cells first, so they got this one single cell population, and then wanted to then illuminate the intestinal crypts and then everything else in between. And you're able in one ROI to compare the gene or protein expression of these three different cell areas, cell types. And if you're doing, like I mentioned, we're now working with groups looking at a population of astrocytes in one part of the brain, multiple parts of the brain, and moving on from there. And then they were able to visualize everything with heat maps, clustering according to tryptase and pancytokeratin and connective tissue, and then also the crypts of the epithelial regions being resolved into t-SNE and the PCA plots, showing that the ability to separate those different ROIs. I've seen a lot of brain examples today. I try to focus a little bit more on cancer examples, but since there were so many brain ones, this is a really exciting, it's still in a preprint out of by Kenny Roberts, who's been working with Oma Bayrakhtar of the Wellcome Sanger Institute. And just a nice way to show how they drew their ROIs into different parts of the brain. But the part that really excited me as well is the reproducibility. So they took us through the paces and showed a very impressive biological reproducibility, technical reproducibility. So what this shows is we are a technology that you can do one slide with a TMA on it. You can do multiple slides. If you've got large projects with many cohorts, our technology has the reproducibility and the throughput to be able to handle that. And I mentioned it earlier, but reproducibility, it turns out, makes data analysis easier. This is important, and we're really excited about how good the data was coming out of the Sanger Institute. We also had some interesting stuff come out of the Broad Institute. This was with colorectal carcinoma. This was a group that had a whole lot of early single-cell RNA-seq data. They were an early adopter of our whole transcriptome, which has now been released. But what I liked about this is they had a CRC tumor sample and drew some ROIs in low-grade tumor, some ROIs in high-grade tumor, and then lymph node metastasis, some ROIs there as well. The pathology looked really similar between the lymph node metastasis and the low-grade core tumor. But when you now can get to the whole transcriptome level, they found an IDO1 cluster that was very similar to the high-grade tumor, thus giving them an idea of where this metastasis had budded from. And then if you want to do a grid pattern, we of course can do a grid. So if you're doing that type of gridded discovery work, it's quite trivial for us as well. This is an example where we did it on lung, 200 some odd ROIs. We're able to also segment inside if you want for tumor and non-tumor. But what's special about this is we can now deconvolve the gene expression data to give an accurate estimate of the cell subpopulations in each ROI. So that's what these single cell RNA-seq groups are so excited about, to now give that spatial information back into the tissue. You can see the cell subtypes that were found. The one way to visualize that with this particular pie chart. And then what I'm seeing a little bit more commonly is using these large or small circles to indicate the number of that particular cell type, and seeing it spread where B cells were found in proximity to the T cells, but the T cells maybe were out in different areas, macrophages being found in different regions. And then everything with all the cell subtypes were found, all of that can be done and visualized accordingly. And for every ROI, showing you the clustering, different ways to play with your data, and different ways to simplify that data so that it's manageable. Of course, we can export it as a CSV and images. If you've got access to these biostats groups and they have time for your data, great, not a problem. But we work hard to also make it manageable for groups that don't have access to that or don't want to go and have that much to look at. And we think it gets you to publications quicker. So the last slide is just an overview. FFPE, fresh frozen, RNA and protein. The tunable ROI selection adds a lot of value if you want to look at one cell population, or if you want to look at ROIs exactly where you're drawing them to enrich for a certain area and compare it. And then of course the reproducibility to be able to do that very accurately, slide to slide. Then the throughput to do that. And of course, the plexing, 18,000 genes for RNA whole transcriptome for that discovery work. And then if you want to translate that into protein, we can do 80, 100 plex with protein. And thank you.